Welcome everybody again to another episode of the ideaXme show that profiles the humans behind the really big ideas that are going to be shaping our world, inspiring the future creators, and for all those that like really great stories. I'm Ira Pastor, your health, aging, and longevity ambassador with you today. So on the last several shows, we've been spending a lot of time at what I refer to as sort of the different hierarchical levels of the architecture of life, disease, and aging. We've talked a bit about the genome, the microbiome, systems biology, tissue engineering. One very crucial area that we have not ventured into yet, which is where we're going to be going today, is into the area of the extracellular matrix. The extracellular matrix, or ECM as it's called, is a non-cellular component that is present within all tissues and organs of the body. And it provides not only essential physical scaffolding for the cells and their constituents, but is also extremely important in this crucial biochemical and biomechanical signaling required for tissue morphogenesis, cellular differentiation, and homeostasis. In each of our organs, the extracellular matrix composition is distinct, and it has a very unique and active 3D structure that is formed by a network of various macromolecules, including collagen, enzymes, glycoproteins, and so forth, that provide this structural and biochemical support and is continually undergoing remodeling and changes to regulate tissue homeostasis, growth, healing, injury repair. And when it's impaired, things go wrong, and it can play a role in many diseases, including chronic obstructive pulmonary disease, pancreatic ductal disorder, spinal cord injury, metastasis in cancer, and even in the area of neurodegeneration conditions in the brain, such as Alzheimer's. And really a greater understanding and comprehension of the way the ECM works and regulates organ structure and function and how this remodeling occurs is really going to be crucial for the development of new treatments and cures for disease. So for today's guest, I'm extremely excited. Joining us, we have Professor Dr. Collin Ewald from the Department of Health Sciences and Technology at ETH Zurich, the Swiss Federal Institute of Technology in Zurich. He is also the founder of the Swiss Society for Aging Research and the vice president of the Swiss Society for Matrix Biology. With a master's in molecular biology from the University of Basel and a PhD in neuroscience from City University, New York, Dr. Ewald's career has taken him through many prominent research organizations so far to date, including Harvard Medical School, Joslin Diabetes Center, Whitehead Institute. And Dr. Ewald's group at ETH studies the molecular mechanisms that prolong health during aging, using C. elegans as a model, in order to develop novel strategies to treat age-related pathologies. And their recent work has shown that many health and longevity-promoting interventions technically reactivate the expression of various extracellular matrix genes. He's extensively published in this area, and most recently his group published a fascinating paper on his work entitled The Matrosome During Aging and Longevity, a systems-level approach towards defining matriotypes promoting healthy aging. And the matrosome is defined as sort of a battery of genes that encodes the ensemble of extracellular matrix associated proteins. It's going to be a fascinating discussion, fascinating topic. Dr. Ewald, thank you so much for joining us on the show today.
Thank you for having me. And
Absolutely.
Thank you for a very nice introduction.
Thank you. I would like to, give you the floor for a while, just so the audience can learn about you, your background, where you grew up, ultimately how you got interested in health, how you got interested in aging, neuroscience, and ultimately, your path into this, what I call extremely important and fascinating area of biology and sort of what makes the 30 trillion or so cells that make us all up, really work, and that is the extracellular matrix. So please, tell us your story in this front.
Thank you. If I would summarize the whole story, I would say all I do, I just follow the science I'm doing. And so that's the summary. But basically, I was born and grew up in Basel, Switzerland. And at the University of Basel, I had lecture courses by Professor Michael Hall, and he was talking about the toll kinase, right? He's one of the founding fathers of the mTOR kinase, which is important for growth and of course for aging, right? And so I was fascinated by that kinase, and at that time we still had to go to the library, so I spent an afternoon in the library looking through random papers with mTOR, and I stumbled upon a paper that looked at mTOR activity in C. elegans. And it was around, think 2001, 2002, by a group here in Switzerland, Fritz Müller in Fribourg. And what they've done, they reduced the activity of this TOR kinase in C. elegans and were able to double C. elegans lifespan.
Mm-hmm.
And so this blew me away, right? Because you just inhibit one gene, you can double the lifespan. And I started looking around, well, has that been done before? And of course, Cynthia Kenyon and colleagues in 1993 looked at the DAF2 gene, which is reducing insulin IGF-1 signaling that can double the animal's lifespan, again, in C. elegans.
Right.
And this is just perplexing how, a single gene can just, if you inhibit a single gene or reduce the function of a single gene, it can double the lifespan of an animal, right? So 20,000 gene, one is reduced, it's a doubling in lifespan, it's one nucleotide change.
Mm-hmm.
And what Cynthia Kenyon further has shown is that this is under genetic control. So a second gene can completely abolish this longevity. And the second gene is a transcription factor, FOXO or DAF16 in C. elegans. Now, I was lucky, I learned that a postdoc from Cynthia Kenyon joined the FMI in Basel. And so she opened her lab here in Basel. So I asked her if I could join her lab and work in her lab. And there I got fascinated how neurons actually regulate the rate of aging. So what this postdoc, former postdoc Joya Seeders, if she ablated single neurons in C. elegans for taste receptor or smell, that could not double, but it could increase the lifespan or shorten the lifespan, right?
Okay.
So suggesting that long lifespan is under neuronal control. And at the same time, there was a study coming out from Harvard, I think, or Harvard Medical School, where they had students to meditate for 10 minutes each day for 6 weeks, and they took their blood, and that changed actually the gene expression in their blood. So basically, I mean, it's a big leap, but basically what I was thinking is, well, thoughts are produced by your neurons, right? And it can change gene expression. And now in the simple organism, you just lose one of the neurons and you can alter the lifespan. So that made me curious to figure out a little bit more. And I decided to do a PhD in neuroscience and went on to New York to do it with Chris Lee or Christine Lee.
Okay.
She also used C. elegans, but she looked at Alzheimer-related proteins. And so my PhD was focused how a human disease is kind of tried to be modeled, not to be modeled, but try to understand the fundamental basics of this proteins in C. elegans. And from there, I really learned more and more about aging research itself, right? Because, if you could stop just one disease, that would be great, but you would get something else like cancer or other things. So then it really suggested to go hardcore into aging. And the thing I told you with Cynthia Kenyon before, that, these longevity interventions are under transcriptional control with transcription factors.
Sure.
Made me look around where to do my postdoc lab, right? And FOXO DAF16 has been very well studied, and many groups study this. So I decided to go to Keith Blackwell's lab at Harvard Medical School, who was looking at SKIN1, which is the NRF2 homolog. It's an important transcription factor for the oxidative stress response.
Okay.
And at that time, they just shown that this transcription factor is actually phosphorylated from insulin signaling pathway, right? So it links insulin signaling to this transcription factor. And so when I was a postdoc in Keith Blackwell's lab, I looked how reduced insulin signaling increases lifespan via this transcription factor. And basically what I was even more interested in is what are the genes that are transcribed under these conditions, right? So I looked at microarray or RNA-seq data, so basic gene expression, always comparing long-lived animals to normal-lived animals.
Okay.
To figure out what are the genes that are actually, upregulated, or in other words, which mechanisms are mobilized under these conditions. And so when I look at cross-expression profile, this meaning just reading all the lists, right? And looking what are the most operated things. I mean, lots of things like better protein homeostasis came out, better oxidative stress resistance. But one striking thing was always on the top, and these were collagen or exomatrix protein, right? And so I didn't know what this meant. Actually, at first I thought this is just an artifact. But if you compare any intervention that doesn't lead to lifespan increase, these collagens are not really expressed. And so I thought there might be something to it. And so to test this, C. elegans is a great model because it lives for 3 weeks, right? Sure. So I took long-lived mutants of C. elegans that have to double the lifespans, and asked whether if I knock down individual collagens in these long-lived animals, would that block the lifespan extension of the benefits? And it actually did, to a very good amount. And that was surprising. And the second surprise was this really suggested that these prolonged expression of these collagens is required for longevity. And the second surprising thing was that if I overexpress certain key collagens, it was sufficient to increase the lifespan of the animal. So if something is required and sufficient, it suggests some sort of mechanism. And this sort of mechanism we're trying at the moment to untangle and figure out. Like the basic question is, under these long-lived conditions, why would these animals invest in exomatrix proteins like collagens? I mean, this is a huge investment. These are big proteins, right? They're synthesized by the cell, they're secreted, they're laid in a network. And we are trying to get our foothold how this works on a genetic level and how this, how can we manipulate this, in other words. And so basically what I found is, there are lots of mechanisms known that protect organelles or proteins or macromolecules inside the cells. Mm-hmm. That's what I found is that, these animals or these organisms also invest in proteins outside of the cells. So in other words, how are these proteins maintained during aging, or, how they maintain to have a functional function of the cells then.
Yeah, I've always been a very sort of big proponent in understanding the various hierarchies that each level of life, let's just say, DNA to cell to tissue to organ and so forth. Everything sort of reports to something else in the sense, and this bidirectional sort of communication fashion, and you have this cell extracellular matrix relationship, which is extremely crucial. Yet, you think of us when we have 30 trillion or so cells, 200 so different cell lineages that make up our bodies. Without the extracellular matrix, nothing happens. Things go wrong. Could you just talk a little bit? I gave a little introduction, but could you sort of, just for the audience's sake, just give a little bit more or sort of insight to the extracellular matrix as a player in this relationship? Because I think everyone is very aware of cells and proteins in the genome, whether you're a scientist or not. But most people get kind of shocked. Wow, there's this other thing. It's not our skeleton. It's this other stuff that is throughout our body. It's skeleton-like, but really the importance of this higher system that sits above and around the cells that is extremely important just in general in our daily life of keeping us whole, let's say.
Exactly. I mean, so if you think about, us, we are multicellular organisms. So we have, as you mentioned, 37 billion trillions of cells, right? And they have to all be held in place and work together, right?
Sure.
And so they're sectioned into organs and things like that. So organs itself, they have so-called the basement membrane that surrounds them, right? So they hold them in place, and this is important. But then cells have also, it's called the interstitial exomatrix, that our cells are embedded into the connective tissue, right? And so so what you have to understand, there is a really strong intercalation between exome matrix and the cells, because the cell makes the exome matrix, but the exome matrix is tuned to the cellular needs, right? Mm-hmm. So the cells, they pull on this exome matrix, they are a little bit anchored, right? And if it's too loose, they make a little bit more. If it's too stiff, they try to express enzymes that cleave it a little bit, right? So they're really well anchored, they're in there, and it's very necessary. So if you look at electron microscopy picture through sections, right? Right. It's very hard to tell whether you look at the inside or the outside of the cell if you don't look at the organelles, right? So inside the cells, you have the nucleus and you have organs and things like that. But inside the cells, there are cables going through, like actin and tubulin, that hold everything in place inside the cells, and that changes how the cell moves. But then you have a cell membrane, right? And then there are these little receptors that anchor it. But then out there, the cables continue, right? It's like a continuous, right? And these cables are then collagens or elastins or other things, right? And so this is really well connected. So basically, in the exome matrix, these collagens, you pull there, it will go over these cell surface receptors into the cell, this pull, this force, and it goes through actin and tubulin all to the nucleus. And so you pull outside, you pull at the nucleus, and you can change gene expression there, right? So there's a very intricate link, and so they come together. So even if you would do cell culture experiments, if you detach cells from the surface, they just die, right? Mm-hmm. So cell attachment is very important. And so this, what you see during aging, right, is that you see some tissues have cellular loss, and some tissues have lots of loss of collagen exome matrix. So we see this best in skin, where we get wrinkles and skin sagging, right? This is due to collagen loss, but at the same time, cells that attach to those collagens, they detach, they round up, and they also, get eliminated or die, in other words, right? And so if you break down the exome matrix, and that's happening during aging, that it gets fragmented and broken down, that loses the cell attachment. And then if that's lost, the cell is lost. And so no new exome matrix is produced, right? And so it's like a vicious cycle going downwards, right? So it's hard to say what comes first. Did the exome matrix integrity is lost, or cellular homeostasis is lost, right? So one leads to the other, and it just trickles down. Now, since there's so much intercalated, you could either change things in the exome matrix that would reprogram cells, or vice versa. So one example is, if you look at cells that stop proliferating, these are called the senescent cells, right? It could be due to the Hayford limit, how many times a cell can divide. Now, you take those senescent cells, they cannot divide anymore, they sit there. You take them and you put them into a young exome matrix, right? So you have an exome matrix that you decellularize, you take all the other cells out, and you take those old cells in the young exome matrix. That completely reprograms these old cells to start to proliferate again, right? It's like a rejuvenation effect. Now, these papers have been published. At the moment, the understanding is we don't know why that is happening. It could be that the exome matrix has, of course, also growth factors and other reservoirs that are in there, right? So maybe in the young exome matrix, these hormones, they're still in there. They, with the cell contact, they can reprogram. Or there's something else to it. And so this is one of our basic questions, what we're trying to figure out, what constitutes a young exome matrix. Right? And in order to understand that, we need to know what are the proteins that are actually in the exome matrix. And so there has been a definition, which I like, by Alexandra Nappa and Richard Hines. It's called the matrisome. So these are all the proteins that in a genome get encoded, that are secreted and integrated into exome matrices. So there are around 300 of these proteins. These include collagens, protoglycan, glycoproteins. So they form the exosomal matrix itself. But in order to form the exosomal matrix, you need also enzymes that remodel it. You need things that associate with the exosomal matrix, like other proteins. And also there are lots of growth factors that are just secreted out there and they're stored in like a growth factor reservoir, right? And so these things are called the associated matrisome.
Okay.
And in total, these are around 1,000 proteins, which is out of the 2,200 or 500 encoding proteins in humans is roughly 1%, is roughly 4% of the total genome, right? Mm-hmm. And so this is very nicely established. So we know this matrisome or all these composition exosomal matrices in theory, what is out there for humans and for mice. We recently with Alexander Napper defined it also for C. elegans, and there's around 700, which is also roughly 4% of the genome of C. elegans. There are around 20,000 genes, so it's not that different. Mm. And now the trick or the thing becomes now to figure out now what constitutes a young exosomal matrix. And as you mentioned in your introduction, the striking thing is that every cell type or every tissue produces its own extracellular matrix. So the composition looks different, right? And this composition that's associated with the cell type, right? I coined the name matriotype for matrisome to matrisome associated phenotype or cell type or disease type. And so what's also clear is that different disease types are associated with a different exosomal matrix. So tumor or tumor severity, you can actually type by looking at the composition of the exosomal matrix.
Sure.
And so the idea is now to figure out what composition or matriotype has the exosomal matrix doing aging in different tissues, right? So how does this change, right? And then once we know there's longitudinal, we're trying to figure out how is it different in long-lived animals, right? If you have mutation or an intervention that doubled the lifespan, how does it look different? And then you can try to figure out like, okay, now we know what's different. We have everything defined. We know what's different. Now we're trying to manipulate it to get it in this direction. And so that's the basic idea we're trying to work at the moment.
When you talk about it, there's so many different angles to come at this. Some from a sort of a thinking about therapeutic interventions at some point. Obviously, the development of small molecules or other forms of drugs that can reprogram or modulate the different types of extracellular matrix expression, but then you also talk about sort of what is a young extracellular matrix. And it reminded me, it's a couple of years ago now, but there was a group, I forget exactly where, Cornell or something like that, where they were experimenting, some of these regenerative organisms, amphibians and zebrafish and stuff like that. One of their unique qualities is they have this flexibility, let's say, in extracellular matrix remodeling versus humans, where they can, make their extracellular matrix a little more, I'll say flexible. I know that's not the appropriate terminology, and take on certain ECM properties of fetal development or whatever. So, I see sort of both. One thing that's a third area that we hear talked about a lot in the aging longevity space is sort of this topic of cross-linking and these forms of extracellular or junk or garbage that connect inappropriately throughout our lifetime to our extracellular matrix and make it stiff or pliable or what have you. Are you doing work in this area as well in terms of cross-link breaking and getting rid of some of these non-ECM things that we don't want hanging around our pristine ECM, let's say, put it that way? Are you also looking in at this particular area?
That's correct. Okay, so I've tried to. It was
a long question. I apologize. Have at it.
Question, right? So I tried to answer the first part. So in terms of trackability,
Mm-hmm.
I think the ECM has a unique advantage, right? I mean, if you think about drugs, I mean, they're the small molecules that can go inside the cells,
Mm-hmm.
and they target around 10 to 50% of the human proteins, right, as a target. And then there's, the biologics, which are pretty big antibodies, are big, they cannot penetrate into the cells, and they usually act on the cell surface. And again, here we have 10 to 15% of all the possible targets we have there. Now, if we use, for example, this biologic, if we understand how the outside communicates in the inside for the reprogramming, right? All we need to understand there is or identify those targets or those pathways. We could do these biologic to target that part, right? So in other words, I mean, there are these cell surface receptor like integrins, right? I mean, there are different antibodies already against them, and they're used in different diseases, right? Because if you can
activate
or inhibit them, that trickles down to different gene expression and changes that. And I think if people have done going into this cross-linking or ECM stiffness as a therapeutic avenue. So it's called mechanomedicine is the term there. And if I'm correct, I think there are 11 different targets that are being addressed there, from TGF-beta to these integrins to other players in the exome matrix, with the end term to lessen the stiffness or increase the stiffness of the exome matrix. So usually it's lessening the stiffness, right? So it's less compact. And I think there are, over 20 clinical trials on them for different diseases. And so I think I'm going to skip the second part with the rejuvenation, because I don't know much about it. It's very fascinating, but there I have to read up. So I'm clear.
We'll do that on another show, no problem.
Another show if I know more about
back
it.
for that one. You have to read those papers.
It's fascinating, but that leads me to the third one, to the glycation end products and to the cross-linking. Of course, yes, we're heavily investigating this at the moment, and so to understand this, so collagens themselves, they form these fibers, right? They are like there, it's like a fishnet, and they can get cross-linked between them. And usually the cross-link is a sugar added to it. So these are these glycation. It's non-enzymatic. It just happens. It takes months to years for the sugar to add, right? But these sugars are usually added to arginines or lysines, and then with time they can get cross-linked. And these cross-links make the exome matrix more stiffer. And so for diabetic people, this is a huge problem, because if you have more sugar in your blood, right, it goes to your tissue and it just drives this glycation and this cross-linking. And so diabetic patients have problems with wound healing, right? There's these ulcers at the foot, for example, that never close anymore, and that could be directly connected to the exome matrix, to this cross-link, right? And so that's why we were also doing this at the Charleston Diabetes Center, where we saw also many of these patients. And so so these collagens can get glycated and cross-linked, right? And so one thing I left out is that I'm talking about the exome matrix turnover. And so the turnover only works, of course, if there is a cell close to it, right? Otherwise, it cannot be turned over, because the cells has to produce the enzyme to cleave out whatever is glycated or cross-linked, right? And then take it in and degrade it and produce new exome matrix to make the turnover. Okay. But we have tissues where this is not the case, right? So, for example, in the islands, the collagens are produced once and they are there for your lifetime. So the half-lifetime of the collagen in the islands is about over 117 years, right? Mm-hmm. And so you can imagine this is then a long time, and then lots of cross-linking in this glycation can happen to those. But usually, tissues that are, lots of cells around, like your Achilles tendon, for example, if you go running, within 72 hours, new collagens are produced and the turnover starts. So, these are the two differences, right? And so the question now becomes, can we if you accumulate these collocation and cross-linking, can the cells of the body repair this out or turn this over, right? And so we have some tendernizing evidence. Actually, other people have done that in long-lived mice. So if you look at mice, there are also these dwarf mice that live extremely much longer than wild type, and you can take the collagen from them. So usually that's done by using these tail tendons. So the tail, there's the tendon, is basically pure collagen, right? So you take those ones out, and you can take these tendons and attach a little weight to it. And you put this in a bath of urea to denature the proteins, right?
Okay.
And in 30 minutes, this weight is it pulls on it, and in 30 minutes, this thing is gonna break, this tendon, and the weight drops. And so if the tendon is young, it takes 30 minutes. When the mice are older, like 2 years old, it will take around 3 hours, I think. Yeah, it's 3 hours. So it takes much longer. It's probably because these collagens get more cross-linked and they hold better together. Now, the fascinating part is in these long-lived animals, even if they're really old, they break as fast as the young collagen tendon, right? And so this really suggests that these long-lived animals, right, or these longevity interventions, somehow invest or remodel these collagens, right? Either they protect against this Caucasian cross-linking or they remodel it. And so since we look at the gene expression level and we see that these gene expression, these collagens, newly ones are produced, we think this is a turnover going on, right? And so it could be technical. If we could like activate this turnover again, in older mice or older people, that would be fascinating to see where we can remodel out these cross-links. Right?
Absolutely.
And so that's what we're trying to do. So basically, this cross-linking or glycation just accumulates with aging, right? And if it's not turned over, then it just stays there for longer, right? And so with any process, I think this turnover of extra matrix protein works very well when the animal is young or during development, right? But breaks down during aging. So for example, if you go and work out in the gym, your muscle is going to grow, right? But your muscle is surrounded by an extra matrix, so the extra matrix has to be adjusted, right? So yeah, it's the same thing, right? So everything starts to decline with aging, right? And I think these long-lived animals or these longevity interventions, all they do, they just either prolong this homeostasis for longer or they just reactivate it. So we do the same thing also with C. elegans, but these are very preliminary data what we're trying to do. But we use C. elegans as a model for this cross-linking and stuff like that because it's just fast aging. But that's to come.
No, still it's very important work, nonetheless. Looking out, I know this requires a crystal ball or whatever, but if the issue with obviously aging for everybody involved in the space is it's very difficult to develop products specifically for process of aging, hence the need to develop different therapeutic interventions at the disease level. Looking out 10, 15 years, taking your technology and moving in the direction, what are you most interested in therapeutically? Obviously, I'm a neuroscientist by training. Is it Alzheimer's that you're most interested in? Are there other shorter term therapeutic interventions that you foresee? If a bag of money of infinite dollars fell from the sky and you had the ability to develop a variety of therapeutic interventions based on your research 10, 15 years out now, where will you take this? What are the most interesting things for you to go after therapeutically?
Therapeutically, I mean, if I had tons of money and
if you had unlimited funds right now.
I mean, the romantic hope is actually to find an intervention, right, that would increase the health during aging, right? And that would affect all those diseases, right? And so clinical trials take a long time. So if I had the right target, and tons of money, I would set up something in that direction, which is still hard to prove how we could show actually that there is a slowing of aging or an improvement or rejuvenation. So we also need some biomarkers. But I'm really hopeful with these matriotypes, right? If you know how they look at during aging, maybe you could use those ones as benchmarking, saying like how old it is, right? So we have the epigenetic clock, so to speak. Sure. Maybe get the metabiotic clock, so to say, that we can say that correlates with chronic or biological age. Yes, I mean, anything that would work. It's so hard, right? Anything. If it could make an impact in neuroscience, I'd be super, super happy in Alzheimer's, right? I'd be super happy. If it makes an impact in arthrosis, I would be super, super happy, right? Even if it just improves life quality of people, right? That would make me happy, right? If they, they don't lose the mobility, they become less frail. That would be, it's just life quality for people, right? I mean, everybody ages and everybody knows how it looks like when you get older, and some people stay very healthy and mobile when they're old, and others ones, they're just fragile, right? And cannot move anymore. And the hope would really be like to shift to the, being healthy kind of thing. Sorry, I just have no.
No. That's a great answer. I think the important thing that comes out of that is that this type of work, extracellular matrix, it's everywhere. It's around all 50 trillion cells, 210 or whatever different lineages. Your work can impact everything. It's, this is not something that only will work for diabetes. So I think it's a beautiful and elegant thing about what you're working on. And I think that's extremely important for everyone to realize, because extracellular matrix is, I love the topic, but maybe it'll be lesser known to some people now, but people should know about it. And I think it's extremely important what you're doing in pushing it forward and, increasing this understanding about all the possibilities, which is great. I mean, it's extremely important. We're at that point of the show where we typically push the science and the story aside, and we get a little more into science fiction, but I'm going to pose the question to you that we pose to every guest, and take your time with this one if you want. Obviously, you've met a lot of fascinating people throughout your career so far, scientists and so forth. This next question has to do with who Professor Dr. Collin Ewald would have liked to meet. It doesn't have to be a scientist. It could be. It could be an artist, a philosopher, a writer, somebody from the past that if you had the opportunity to meet them, Einstein, Edison, whomever, Professor Dr. Collin Ewald would like to sit down with and talk to them for a little bit about any topic. For you, who would that be?
This is a very tough question, but I knew it would come because I know your show. Mm-hmm. And by the way, I love your show. I mean, it's just, it's a great show. And I've been thinking a long time about it. I mean, people I admire from the scientific standpoint, for example, George Church, you had an interview there, right? Mm-hmm. I met George Church when I was in Harvard Medical School, but it's also like I like these interviews because you can figure out and see how their brain works, how they answer questions, how they address a certain problem, right? Mm-hmm. And so this is exactly what I think I would like the most to meet from a scientific person. And so I've been thinking a long time, and I would say Sidney Brenner. It's not just because I see elegance, but Sidney Brenner, and it's a specific time period actually, because Sidney described the time when he shared an office with Francis Crick, right? And they were discussing wild ideas, but really like harsh, right? So that's what I've seen or heard from the interviews, that somebody proposed an idea and they go back and forth, very, very hard thinking. So I wish I would have been there and tried to see whether I could keep up with them, right? The intellectual battle, attacking a problem. So I think that would be like, the place and time and the persons I would love to meet.
Excellent answer. Once again, I really want to take the opportunity to thank you for joining us today and sharing your knowledge in such an important area of, once again, what I refer to as the biologic architecture of life. Thank you for everything you're doing in the lab and moving sort of the human story forward, and moving the story of health and longevity interventions forward in this area that, frankly, I think needs a lot more attention. And hopefully we'll be able to spread the word and get it out there. So once again, everyone listening and watching this show, we've been talking with Professor Dr. Collin Ewald. He's at the Department of Health Sciences and Technology at ETH Zurich. He's also the founder of the Swiss Society for Aging Research and the vice president of the Swiss Society for Matrix Biology. Heavily suggest you pick up his most recent paper, The Matrixome During Aging and Longevity, a systems-level approach towards defining metabolites, right, promoting healthy aging. A clearly fascinating area of biomedical endeavor that is a major part moving forward of solving this health and longevity issue. And Collin, once again, thank you so much for taking the time out of your very busy schedule to join us today.
Thank you so much. It was a lot of fun.